All right. Hello. Thank you, everyone. All right. So today I'm here kind of twofold. I want to talk about the EveryCure sort of organization, and essentially this actually serves as basically an extension of Tyler's presentation from earlier. I'm going to try to leave a lot of time for questions and discussions as much as I can. So I'm going to try to keep things pretty brief. But basically, the EveryCure project is a nonprofit that was founded about a year ago now, essentially trying to tackle the drug repurposing problem. So I'm going to cover basically that. So right now there are 3,000 FDA-approved drugs approximately, and each one of those drugs is essentially only approved for one explicit use, and it's FDA labeled. That is a bit of a muddled definition sometimes. It can get very specific, but that's the general claim. So sort of the idea underlying everything we're trying to do is that these 3,000 drugs can be repurposed at low cost and very high efficiency to help patients. So my PI, David Feigenbaum, actually works at a Castleman treatment center, and they have found over 10 repurposing treatments for Castleman disease and a broader set of cancers. Specifically, a treatment for angiosarcomas is a good example of the kinds of work that gets done. Was uncovered in 2013, but was not thoroughly explored until 2016. So they were able to find that this angiosarcoma patient was actually treated by a PD-1 inhibitor that was previously unknown. And so by sort of taking this knowledge that was in the literature and applying it more broadly to the biomedical patients, they were able to achieve their goals. So this is the world that we live in right now. There is diseases that have treatments, and then there's essentially like a barrier that of diseases that won't have treatments. Further reflecting off of Tyler's point from earlier, 30 million Americans are expected to have rare diseases. That number may be higher. We are actually uncovering more rare diseases every day. So we want to try to start developing mechanistic links in existing drug disease combinations that might be able to help us attack some of these issues. Very obvious question, why aren't, this seems like a big problem, why is industry not tackling this? So there's not a lot of money in drug repurposing. As soon as a drug falls off patent, that's it. You get 7 years. You got to optimize the use. If you, in that 7 years, find out your drug can be used for something else, like 2 or 3 years in, that's good. You get like 5 years. If you don't hit that point, it's just, there's no financial gain. There's a lot of risk. There's side effects that for one patient cohort does not affect another patient cohort. So even if you make a drug that works really well for one thing, proposing like an FDA label change for that might expose you to liability, and it also takes a long time for these trials. So the EveryCure org hopefully tries to use the nonprofit status as a way to sort of leapfrog over these issues. We don't care about how long we have to wait. We don't care about the fact that we won't make money because nonprofit, we take a drug that was approved back in the 40s, and it's all the same to us. So this is the world we're trying to create. Just because of the Castleman nature of our group, here's how sirolimus was used to go in my PI David. So they were able to identify mTOR as a potential candidate. Using that mTOR activation, they were able to propose that sirolimus, which is an mTOR inhibitor, may be able to be useful in this drug. They were able to essentially use this in a patient and then evaluate in clinical trial, and now they're trying to get a treatment guideline change proposed. Similarly, a very equivalent pathway was done with rituximab, which is another viable Castleman treatment. This works in as a JAK inhibitor, completely different mechanism as to why Castleman is caused in these ways, but now they're working on doing this in clinical trial. And just sort of to reiterate my point, Siltuximab to develop cost $100 million and was a very expensive treatment, but to repurpose the bandwidth was only $3 million, and it's very much cheaper even on the patient level. So broad timeline, broad plan of here's what we're trying to do as an organization: identify drug disease links, evaluate those links, place them in a clinical trial, and then try to update FDA labels once those kind of get done. All right, so two very broad high-level questions. Why is automating drug repurposing hard? And how do we go about sort of doing this drug repurposing? So first off, why is it hard? And I think this is a very important question. I think it's something that we often kind of skip or overlook, but I think it's important to talk about. So the examples I've given before, and a lot of the examples that we discuss of drug repurposing, are a lot of human expertise that's put into it. So people in general are very good at integrating complex sets of information. These biomedical experts have access to legitimately sometimes half a century of experience when making these decisions, sometimes more, plus all of the teachers they've had, all of that expertise. If you get a room of 30 very, very high-level experts in a room together discussing, that can be like hundreds of thousands of man-hours of experience. And a lot of these decisions they come to actually are made subconsciously on an intuitive level. Medical scenarios are even more so prone to this sort of very subconscious bias. It's actually very hard for people to explain why they make the decisions they do. They tend to be it tends to be very just difficult and much more based upon just having an understanding that something will work than an actual legitimate biomechanical reason. And that's actually really good. That works really well a lot of the time. But modeling the underlying reasoning, or how humans figure out these drug disease relationships, is almost it's a very broad problem that I think we've only sort of dipped our toe into. I want to sort of pin that idea there. So a question is then, how do we go about doing this very hard task? So major methods of computational drug repurposing it kind of positions it as a chemistry problem. What chemical structures of previous antibiotics can be used to predict a new antibiotic? What drug will specifically bind a receptor? And so this kind of rely upon exact disease mechanistic knowledge or previous drugs which already do something that we want to do. The problem, especially in the rare disease space, is that these mechanisms are generally unknown, or in the case of Castleman, even I mentioned a few slides ago, there's two completely different mechanisms for this disease, and targeting one of those using a computational method will completely ignore the patient cohort affected by the other mechanism. So continuing on, the best approach we have so far is trying to use a knowledge graph, essentially, to model this high level interconnectedness of biomedical data. So sort of, again, extending off of Tyler's talk, the large scale modeling and hypothesis generation has been taken on by the NCAT's Biomedical Translator Consortium. And a knowledge graph provides us a way to generate these computational hypotheses when we have imperfect knowledge or limited information. So rare diseases where you maybe have a statistical association with a gene, a knowledge graph can provide you an explanation or a couple potential palliative drugs for that scenario. I want to sort of go into not on a too technical of a level, but there are 5 groups within the translator, 5 ARAs, who have tried to do this drug repurposing problem. So the first one is the RoboCop group. They use rule mining to essentially find patterns in their graph, which they say, historically, if a drug is treated by if a disease is treated by a drug, we see it goes through a gene, or it goes through it gets down regulates this other pathway, and then this system gets really activated, so it's able to find those kind of meta patterns automatically. The Medi-Canron system tries to count the number of supporting publications and gives you a score between 1 and 0. This is hopefully going to be, I've talked to Will a bunch about this, and really looking forward to the next version, but it works well too. BTE does they do this normalized Google distance calculation, which is essentially a co-occurrence comparison between how often a specific drug and disease occur together amongst all of the different databases that underlie BTE. And that tends to produce pretty interesting results. And Spoke does this very interesting building up pathways of facts in their knowledge graph, and then they're able to incorporate the EHR data from UCSF, and then from that result they are able to use PageRank to get drug disease scores. And then last but not least is the ARAX methodology. This uses a drug embedding methodology. It's specifically neural net based. They mathematically represent nodes, and then they compare how similar or different each of those nodes are. So these are 5 very orthogonal approaches to drug disease matching. I think that's one of the major benefits of what the biomedical data translator project has provided us so far, is these extremely different ways they're trying to go about solving the same problem. So what kind of have my team been doing? So all these systems sort of were put out into the world without really any intention of doing sort of what EveryCure is trying to do right now. So we went through and we just systematically asked every single group, hey, what do you know about this drug? What do you know about this disease? What do you know about this disease? What do you know about this disease? That took a pretty long time and it required a little bit of work. I'll get into that later. But for the first time, all of this has been put into one location, and now, at least for a fixed time point, what the translator system as a whole believed about drug-disease relationships is now captured. And we're actively working on finding new information that here's a very, not projecting very well, but visualization of sort of a subset of how we believe that drugs and diseases are interacting. And then furthermore, here's interesting statistics about how many particular MONDOs are returning for each group in the translator project. And in general, most of the teams are not returning that many drugs for how large these things can be, unfortunately. And if people are interested, here are the top 10 hits from every group for which that makes sense. If you want me to go back to these specifically, I can. Most of the time, they're all getting 1s because there's a lot of stuff way at the top for a lot of these. But so here's, for the first time, something we can do with this. We can look at, here's what all of our groups think will treat influenza. And we're able to actually take this, instead of just running this query, we can actually take all of this in a bulk form and start doing this kind of high-level analysis of it. More interestingly, I think this is very cool. You can just run this for the plague. The Black Plague is a valid MONDO term, and you can just run it in there. And it turns out that the top result from BTE is a plague treatment that will treat the plague, and the one from Spoke is just the plague vaccine. And that got a 1. That actually worked. Spoke did the best. I actually think that is insanely impressive that they somehow Spoke has it in the knowledge graph that plague vaccine cures the plague, and if you go and ask it, hey, what's going to cure the plague, it will just tell you directly. Yeah, it's so yeah, no, so kind of for the first time, we're able to start doing this really fast, high-level analysis of what do we know? What do we know about these things? So there's a lot of issues, and I don't want to speak about these issues in a disparaging way. I think these are things we're actively working on fixing, and I think hopefully my team will be a large part of this. So there's a lot of occasionally arrays are down, or hitting them with tens of thousands of queries all at once tends to break them. I don't know why that would happen. It's very unclear to me. Some ARAs were unable to query MONDOs for which they had information. There's certain sparse or non-algorithmic limits on how many drugs would be returned for specific ARAs, which is something that I think is very achievable and fixable in the near future. I think one other translator issue is that some algorithms could you could customize or tune parameters when appropriate. I think that's going to heavily depend on what algorithmic system gets used. Clearly, the 5 algorithms I've discussed are wildly different, so just saying like a do better parameter doesn't really make a lot of sense. But I think things like the novelty are like clinical viability make a lot of sense. And hopefully we're working to be able to fund some of these groups to be able to work on some of the issues that directly affect the EveryCure project. And just something interesting, the sparsity issue. So we were able to kind of look at how much overlap we found, and it's often very low. So because so many of these ARAs are only returning like 400 drugs, the statistical chance that a drug and disease pair that occurs in one of them will occur in another one is just generally like there's tens of thousands, if not hundreds, or there's about 120,000 drugs that are returned across all of Translator. If you just do like a Bayesian model of that, it's just low. Malaria actually did really well. Malaria has 7 drugs that are returned by everyone. So that's actually the best one we have. One other interesting aspect of this, the XDTD methodology, the black box neural net that the AREX group is using can rank any pair, but the direct trade-off of the fact that it can rank any pair is that the pathway it used to generate that explanation is non-existent. Because it's a neural net, the neural net compresses the information so much, there is no like mechanistic explanation as to why. So if you look at the Medi-Cameron approach, that uses paths through the graph to generate a score. That's exactly how it tells you, hey, I think this thing's really good, is 100% because of a path. So it just gives you that path back. It says, hey, here's why I think that this drug will treat this disease. So it's very interesting to see sort of how these high-level algorithmic changes directly affect our own ability to sort of use the results down the line. So this is the EveryCure timeline. We are currently in these 2 phases of the process. And I really want to point out that this might seem very high level and abstract, and when we talk about it in like a conference like this, it really does feel that way. But these are the top Castleman results that came out of the XDTD algorithm. And the top result, which is adalimumab, which is an anti-TNF inhibitor, was concurrently with this kind of the operation of us running this, was actually being tested in a Castleman patient, and is found to be extremely likely to be a viable treatment. So like parallel discovery, proteomics work being done by the Castleman Center was directly aligned with the top scoring result from the Translator project. So pretty incredible kind of, and very like fortuitous that the timing of that just very, very like interesting that worked out really well. So kind of next steps, working on how to combine these scores into one mega score, whatever you want to call it, correcting the issues within the ARA systems, exploring other ways. So those 5 ways of kind of making these scores, really interesting, but there's tons and tons of almost infinite ways that this could be done. And I think another really interesting question is, we are currently asking how likely is this drug-disease connection to exist. It's modeling it completely as a graph problem. The question, honestly, for EveryCure's sake, would be better served as how likely is a clinical trial for the drug and this disease to succeed. It's a much harder question to model, especially with the graph as the input. But that's sort of the next step in the timeline we were kind of proposing here. So yeah, cool. So thank you. This is the link to the results again that we've kind of been my mouse is on screen. But yeah, so yeah, exactly 20 minutes. Any questions?
Thanks, Daniel. So I wanted to thank you for talking more about Translator, because it's, I can only talk so much about it even in 45 minutes. So I appreciate that you went in depth a little bit on the ARAs. And when we started the program, the goal was to try to leverage as many different methods as possible. And I think we've succeeded well at that, and your data shows that. So I think that's really great. I think my biggest question for you would be, how can Translator, as we're looking into the future, how can we work towards supporting your question of whether a given clinical trial might succeed? Like what do you envision as the important steps for that?
So within the Every project, we have like a couple different things, and Jeff has spoken about this, like the PICO of what a trial or what you expect like a drug disease pair to have, maybe expanding the information we have within the Translator as a whole, or sort of just even just proposing that directly as a question we're trying to go about doing. Because I'm not sure for that question that modeling it as a graph algorithm problem is the correct way to do it right now. That's an open question. Like, I just don't know, because it might be the case that we just have to take results we have now in this big set of data and just say, okay, here's the best 1,000 ones. Let's run this through some other computation. I think it's an open question. I think we probably should talk about it more offline.
I think I missed this. When did you run the queries exactly? March, okay.
Yeah, March into April, mid-April for some of them, because some of them failed and I reran them. So yeah,
okay.
We're fixed into that time.
Okay. I guess one of the interesting things and I guess challenges for this sort of assessment is that Translator is changing.
Yes.
A lot.
Right? I agree. I think that was sort of why there was a hesitancy to gather the data in this way. For my purposes, it's nice to have, but if things someone adds a new data set to their thing in May or June, they are going to not be reflected in the results that I have. And so it kind of needs to be reran every few months.
That's right.
Yeah.
Yeah, we're totally revamping our scoring.
I'm excited to get the new stuff, but okay. Right. It's locked in. I'm not getting,
Great.
Thank you. That was a great talk. I just wanted to say I think this work is really important in terms of kind of establishing and updating that matrix of with diverse models, and getting a good stress test of our RTX KG2 service is a good side effect of that. Helps us load test the system, and we're going to work hard to try and put a little more X in the XDTD. So, thank you.
I know you have the explanatory part, but it's not this. It's post facto just because of the nature of the algorithm. It's not a problem. It's just the nature. It's just how it works. Yeah.
Any more questions?
All right.
I guess there is a danger in a way because all these people working on Translator, including myself, want it to work well and want to show it's working well, right? And so, for example, you showed that the top result from one of the reasoners was in a trial in humans. The same time, a skeptic might say, okay, well, what about the number 2 result and the number 3 result and the number 4 result? So did we just happen to get lucky in that case? You know what I mean? If you looked at all the diseases and all the reasoners and all that, are we only sort of lucky some tiny percentage at a time, or how do we assess that? And do we
Yeah.
and do we know all the clinical trials with all the drugs? Is that something we're always
I think I think about this a lot. I think this really gets into like a very broad concept of like verifiability, because we have 500, or even if we boost it up to 10,000, there's no way 10,000 things can treat any disease. You're almost always going to be mostly wrong with your mathematical answers. It's just definitionally, if you were proposing lots of treatments for something, the vast majority of them are going to be wrong. So I think we can only hope to be kind of right, or like so I think that it's very good to be like, I think this is, honestly, if this was number 10, it would not be as impressive, but it would be just as relevant that we got something that, it was because it's both of these things, so XTTD and the proteomics people figured out that TNF was important in Castleman. That's why this hit on both of them simultaneously. It's just very fortuitous that it was number 1, but if the score was 0.2 different or 0.02 different, I don't personally think that would have affected how cool this was or how useful or interesting it was. Which is why, even in our timeline, a lot of this is evaluate with human experts is like a big part of our kind of, methodology. We want those like top 20 results or top 100 to be checked by like a human, like a biomedical expert. That's kind of, because we're going to be mostly wrong. Yeah.
Okay. And just one more part about the anti-TNF. So was it already known that TNF was associated with Castleman? Was that just kind of like a known thing in the literature that everyone agreed upon, or?
It was moderately known, but it was more emphasized within a specific patient subtype. Okay. So that was kind of the proteomics discovery.
I just wanted to add to your point, Will, that we don't have the information on all the clinical trials out there being utilized right now in the way that we definitely could in Translator. And that's actually the subject of one of the sessions we're going to have in our relay in a week and a half, is how can we better utilize clinicaltrials.gov in order to get at exactly those points. So.
And just one final plug for the relay. I hope you're coming to the testing sessions, and I hope we can incorporate your stuff into our benchmarks.
We'll do my best. Yes, I'm going to rerun the thing pretty soon also. So we'll fix it. Time will start again mid-September 2023. That's when we fixed in time soon. Oh, you're...
One more question finally. So on the topic of rerunning these analyses, what kind of metadata do you need to make sure that you can generate reproducible results from the translator queries you do?
Can you be more specific? Like,
right. So one of the things that's on my mind is that we need to version translator. We need to say this is version 1, and you can go back and put that on your analysis and say it was generated on September 1st. We don't do that yet, so I'm curious if there's other metadata that you need.
0.5.
Yeah.
It's a good point. All the groups have kind of their own. So time important. Maybe a seed. I'm not sure. For some of them, have some stochasticity. Hard to say. I don't know I don't know if fixing the seed and running it, everyone gets a seed 42, and we just say that, and then we use those results. I don't know if that's the best long term. So yeah, there's a lot of stochasticity implicit into a lot of these methodologies that make that a very hard problem. Yeah, that's just kind of the nature of how it is working right now.
One more. Yeah.
Sorry, just one last question. I'm wondering, what is sort of the criteria for selecting these algorithms, and how do you consider sort of the interdependencies and how the similarities, how they work, and how the difference, how would you, I guess, incorporate that into your scoring mechanism? And yeah.
Oh, that's a really good question. I don't know. I should have left this thing plugged in. So the way that the scoring methods were chosen was by the PIs of every group, essentially, who are, pretty much the experts in biomedical knowledge graphs as far as I'd say. So they have a pretty good intuition as to what has worked historically. Mostly it was just, we really want to get some scores, what's something that can work at this scale. So that was the main criteria, was scalability at this point. I think, and we have a lot of people within the translator really pushing for this, an idea of, hey, sort of using the MONDO, all these lung cancers are really well, like for the lung cancers we know, all the lung cancers are really well classified by this group's algorithm. So for a new lung cancer that we don't have a treatment for, we should really like listen to them. So kind of like splitting it into disease space. I think that's a really good way to sort of cluster diseases and then give everyone, how good are you at cancer? How good are you at neonatal diseases? How good are you at like blood diseases? That is one way we will probably end up going about weighting these algorithmic results.
Yeah, awesome. That's answering. Thank you.
Yeah.
So I think we're out of time for this session. So thank you, Daniel.